Ladies and gentlemen, welcome back. This is the 5th video. We're cruising right along, and we are continuing to learn more about DNA. And today, we're going to spend more time looking at how or why you want your body to build the protein it does. So let's move on to a slide 29. Now, this is an in-class activity, but if you happen to be absent, I'm going to go over this really briefly, just to give you a quick advice on how to do it. First up, you want to Google PhET DNA. So if you Google PhET DNA, that'll take you to gene expression. You click there, open it up, you click this option, and then you're good for a moment. Then you go back to the worksheet and you're thinking, well, how do I fill this out? So you can fill this out on a spare sheet of paper, or you can get the worksheet from me, and then you can watch this and hopefully figure it out, right? So you want to try to get 9 observations that talk about different things. You want to talk about what you noticed and learned about positive transcription, this thing called RNA polymerase, and this thing called ribosome. These are all different things that are involved in your body making protein. But you may not know exactly everything there is to know. You know some things already. We've talked about it. It's one of our major problems for the chapter is DNA making protein. But you may not know everything about it, but that's okay. So first things first, though, is you read this initial problem. How does DNA produce protein molecules in the body? Your first step today is to read that question and then fill it in right here in this box. So you'd come up with your own hypothesis of how, if somebody asked you this question, how does DNA make protein? Well, what would you tell them? It doesn't have to be anything perfect. Just what do you think you'd say to them if they asked you that question? Just based on what you've learned in class or what you remember. Keep it simple, keep it technical, whatever you're comfortable with, but in your own words, just make a confident statement of what you think would be a good response to this question, this scientific problem. All right, once you do that, then the next step is just moving on and starting to get some observations from the simulation. And this is what you need to do. First of all, you got to, what does positive transcription factor do? And it looks like it wants you to connect to DNA. There's a single observation there. The positive transcription factor bonds to DNA. Not much else to say. You could say it bonds to a region called a gene, so a section of DNA called the gene. You could say that too. And then let's learn about some of these other things. You got RNA polymerase. You can see when you click it, it indicates a region where it needs to go to the DNA. You take it there, and you could say, well, the RNA polymerase, you could say what it looks like. It looks like a blue egg. You could also say that it changes shape and goes across the gene number 1. You could say it made something too. Made a black squiggly thing. Well, what is that? Well, you could add another observation again. You could say, well, it changes shape and goes through the DNA and makes something called mRNA. What's that? Yeah, it's changing shape and riding along and making a new polymer called mRNA. That's great, but what do these other things do? Well, you got ribosomes, and you can see that ribosomes are supposed to go to that mRNA it made. So here's another observation. Our ribosomes, you can even say what they look like. A weird hamburger bun. Or you could describe it whatever you say. You could say it looked like two golden sections together, and they like to connect, or they want to form a bond with the mRNA, and you send it to one. Here's another observation. You could describe what it looks like when it's working. So this ribosome does a job. Ribosomes bond to the mRNA and scan them, pull them through them like spaghetti. Finally, you could say, well, what did it make? What is this thing? Well, look, it's a protein. So all these observations can lead you to answering that final question on the worksheet, which was, how does DNA produce protein? Well, you just saw it. Rewind the video or go to the website yourself. Just ask me, I'll get you the link. This one right here, and I'll make it available and you can perform it yourself. Or just watch the video over and over again. All right, let's move on. Let's move on to slide 30. So it introduces a word called transcription. And transcription is a word for when DNA helps make an RNA. If you look at this graphic, it shows you just that. This is DNA. And you can see that this DNA is getting converted into an RNA molecule, or polymer, I should say. It's a polymer, RNA polymer right here. Now, this thing called RNA polymerase is an enzyme or a type of protein whose job it is to make RNA polymers. That's where it gets its name. So this protein right here will study the DNA and then build an RNA polymer from it. And it's like, you might ask, why does it make DNA to RNA? Well, it's just a first step the body does to make a protein. the body doesn't just take the code from DNA and make a protein. What the body does is has its DNA, then it makes RNA, and then it makes protein from this RNA. Why it does that, I don't know. Just remember that DNA makes the RNA, and the RNA polymerase protein actually studies the code of DNA to make this RNA polymer. And that whole process of DNA becoming RNA is transcription. Make sure you get this definition for slide 30. The next task on slide 30 is, after you get the definition, is to observe model A, which is what we've been talking about here. And your job is to interpret it, kind of like I've already done for you. And you're supposed to come up with a hypothesis on how RNA polymerase, this little popcorn looking thing, right? How it creates all an RNA polymerase, but you're supposed to explain how RNA polymerase does its job of making RNA using the words chemical bond, nucleotide, and polymer. So you want to use those words in your hypothesis. You want to say the RNA polymerase makes RNA by, and then using these words, chemical bond, nucleotide, and polymer to explain how it makes RNA. Good luck. If you have any questions, just come talk to me later, all right? All right, let's look at slide 31. Slide 31 has a definition officially for RNA. RNA is like DNA, but you can see it's single-stranded. And also you can see that it contains some of the similar nucleotides. It's made up of C nucleotide, it has the G nucleotide, and it's made up of the A nucleotide. So as far as CGA is concerned, it's like DNA. But it has one nucleotide different from DNA. Not only is it just one single strand, it's not double-stranded like DNA, it's a single strand, but it also contains this U nucleotide. This is a new nucleotide, it's the letter U. DNA contains thymine, RNA contains uracil, or the U. So DNA has the T nucleotide base, and then RNA has the U nucleotide base. And you know if you're looking at DNA or RNA based on if you see U or T. If you see T, you're looking at DNA. If you see a U, you're looking at RNA. Go ahead and copy these notes down for RNA or ribonucleic acid if you haven't. And then what you're supposed to do is kind of what I've already done for you. You just look at the model here and compare section 1s of RNA to section 1 of DNA, and then compare section 2 of DNA and section 2 of RNA. So examine section 1 and determine how is RNA different from DNA. So what's the one nucleotide? All right, so what's the one nucleotide over here that's different than DNA? And we already discussed that. That is that RNA has U, DNA has the T nucleotide base. That would be your answer number 1. Answer number 2 says, look at this, and what's the difference between the two polymers, DNA versus RNA polymer? What's the difference between them? And that's what you'd write for number 2. And then finally, looking at section 1s and section 2s, how are they similar? How can you compare them? What's similar about them? And so for question 3, you just write, how is DNA similar to RNA? We've already said how they're different. Now for question 3, you just say how they're similar. Just interpreting this model and looking at the nucleotides and the overall structure of the polymer for DNA and RNA. Slide 32, and this is a in-class activity where you're going to actually be turning DNA into RNA, just like we've already seen RNA polymerase do. We've seen RNA polymerase do it in this model. We've seen RNA polymerase do it right here in the PhET simulation. Now's our chance just to do it on paper as an activity, just to make sure we know how DNA turns into RNA. The question is how do you use the U and RNA? We know A's bond with T's, we know G's bond with C's in DNA, but we have to practice here knowing how to convert DNA to the RNA on the dotted line. We'll do this as a group activity, and different sections, different teammates will do. If you miss this day in class, make sure you come and talk with me so I can help you. But the gene you're working with is the same one we've been working with, the pleasure receptor, this protein right here that's in our brain cells that connects to the happiness chemicals to allow our brain to feel pleasure. All right, let's move on. Slide 34, we shall talk about. This will be an in-class activity. It'll be another statement of understanding. So just wait for this in class to get further help with it. On slide 34, even though it's an in-class activity, I'll go ahead and talk about it here now, just in case you missed the day and you need time to try to complete it. The worksheet will be available in class, so you can just copy down the overall structure in your notebook or on a separate sheet of paper. But first thing is the scientific problem. How are proteins chemically formed from RNA? So how are proteins made from RNA? You may not know exactly, and that's okay, but what I need you to be is a little confident and to generate your own hypothesis here in the center. So how do you think protein might be made from RNA? What's your thoughts on it? So go ahead and write an initial hypothesis here. Make sure it's confident and take a chance, even though you're not for sure 100%. But whatever you think is the case, write it here confidently in the hypothesis. And then your next step is to click on a link that I provide you with, and it'll take you right here. Once you're here, your goal is to, what I would recommend is you can just click translate, and you'll see it do its job. Watch it go. And your job is to make observations. If you feel like it goes too quickly, you can stop and reset, and then do step by step, and it'll go slower. One step, you could write an observation, click next step, write a second observation. But your goal, ultimately, is to have observations that help you learn about how proteins are made from RNA. Just to clarify, this is RNA. This is a single strand. RNA is single-stranded, and you know it's RNA because it's got that U nucleotide, the uracil, okay? So this shows you basically how RNA creates a protein. So as you write your observations of things you notice from this simulation, be sure to talk about what the ribosome is doing, which is this big brown thing. Looks strange. It's a large protein that binds to RNA, but just write things about it, what it looks like, what you see it doing, what goes inside it, like these little structures, these green things, right? What's this thing doing? It's called tRNA. What is its job? What do you see it doing? Or what is this at the end? What are these amino acids? What's happening with them? What goes on to build the protein? So as long as you're writing observations that include ideas about the ribosome, the tRNA, and the amino acids, you're perfectly good to go, and you've certainly learned more about how proteins are made from RNA. And then ultimately at the end, you create a model. Let me adjust this. So you're going to create a model or written to show how proteins are made from RNA. All right, good luck. All right, we're going to go back to the translate here and just let it finish up. So just watch as it goes. And you can use this video if you want to complete the task. You can just pause as you need. But this is a protein getting built. The ribosome uses the tRNAs. And the tRNAs bring amino acids in. Those amino acids bond together, forming an amino acid chain. So this is a polymer because it's a repeating molecular structure, the amino acids are. And then the ribosome takes off, leaves the RNA, and you have a protein there, bouncing around. It doesn't look like much, but in your body it has a specific function on a molecular level to help you. It does something for you. We have slide 35 here. We keep on moving. Now, the whole thing that started these notes were, what's the purpose of protein, right? Why does the ribosome make protein? Why do we need DNA to make it? And once again, I'm just going to constantly remind you that proteins, like this innocent little thing, is the basis of our muscles, of the enzymes that do chemical reactions. It's the basis of the proteins that help pump things in and out of our cells that our body needs or needs to get rid of. Proteins are the basis of the hormones that help us sleep or run from an event, the chemicals and all that happen in our brains. So slide 35 is pretty straightforward. You just write down the definition for ribosome, and then you'll hopefully remember the function of ribosomes and how they help make the proteins that our body needs to do all these important functions, building your body one part at a time. And just remember, all these little proteins and the different functions keep your cells running and keep your body happy and healthy. And all these proteins are different from one person to the next, making you unique. And that kind of brings us back to the purpose, when we're really just trying to understand problem number one, how DNA helps us be healthy and live a healthy life, how DNA makes us so unique from one another, and why we should care about the importance of proteins. All right. That wraps up our fifth or sixth, I'm losing count, of a twisted tale of hard work. Have a good day, folks.